Hello students, this is a video for class plus 2 biology. We are going to discuss today sexual reproduction in flowering plants. And as you know that students, you are in plus 2, so I think you people are very much aware about the various parts of the flower. So basically flower has two parts, that is essential part and non-essential parts. Essential parts consist of androecium and gynoecium, which helps in reproduction, and non-essential parts are those parts which are going to just helping part. They are called as helping parts because they are helping in attraction or protection. So this is called as a non-essential part. But our major focus is on essential part, that is androecium and gynoecium. Today I am going to explain you about the androecium, some parts of the androecium. Androecium is basically having two parts, as you know, that filament and anther. That anther is actually a major reproductive male reproductive part of the plant and flower, and you can say that this anther is actually also called as microsporangium. And this anther basically found in two types. One is your bithecus and second is your monothecus. Whenever there are two lobes of anther, you can say that just I want to show you two lobes of anther. For example, look at this. Whenever there are two lobes of anther, suppose sometime anther is formed like a one lobe, like this only. So that time the anther is having one pollen sac, two pollen sacs inside, and this is attached with the filament. Sometimes these pollen sacs are divided into two lobes. One or two lobes are attached together. Then this one is called as bithecus, and this one is called as monothecus. So the bithecus this anther is actually having four pollen sacs, pollen chambers. You can say that if we will cut it from here, we can see it as a, suppose I am going to cut it and then forming you a pollen sac over here, then you will get a four pollen sac on this one, and two there, and this will be filled with the pollen grains. So this is the structure of a bithecus. Mostly this is most common one. So these are the anther's basic structures. So students, this is also these are also called as microsporangia. Sporangia. You can see this. Why they are called as microsporangia? Sporangia is a plural form and microsporangium is a singular form. Microsporangia, pollen sac are the same thing. So let's start with the next topic of this microsporangia, development of microsporangium. How this pollen sac will be developed. Actually students, for the plus 2 student, these are the starting and basics of the, basically whenever the this flower is a young one, when the flower is very young, at that time the this stamen will start developing into one lobe or two lobe, whatever the condition. Then there is one round mass of some cells are there, and those round mass of cell, you can say that this is a round mass of cell. This round mass of cell is called as archegonial cell. Archegonial cells. Here lots of cells are present, and these cells actually called as archegonial, parenchymatous type of cells. And these archegonial cells later on divide in a various manners. Sometimes, mostly they are divided periclinal division. Periclinal division means they will start from here division, and next cell will be start moving towards the space only. And this archegonial cell will divide and convert into some, you can say, the two types of cells: primary parietal cells and primary sporogenous cells. So students, I want to show you this primary parietal and primary sporogenous cell. So look at this. Look at this structure. I want to show you this flow chart. This flow chart you have to learn archegonial cells are converted into primary parietal cell and primary sporogenous cells. These primary parietal and sporogenous cells are further divided into various layers of the anther. Students, as I told you, anther is this part. Look at this one. This is a anther. This anther will be having two layers. One is primary primary parietal cell and primary sporogenous cell. The primary parietal cell will be converted into three major layers of the anther, that is endothelium, middle layer and tapetum. Okay, these are the three layers. Outer layers not related to an production of sex units, but they are just in protective in nature and nourishing in nature. So these three layers are originated from the primary parietal cell, but the next part is primary sporogenous cell. Sporogenous cell means these cells are responsible for the formation of pollen grains. Now these sporogenous cells will convert into microspore mother cells. Students, these are also called pollen mother cells, that means PMCS, and then they will undergo, and these microspore mother cells are actually diploid in nature. They are having double set of chromosomes, and these diploid MMC cells will undergo meiosis, and then they will convert into microspore tetrads and then pollen grains. This all line I will explain you in detail later on. First we will start with these layers. Then when we will come to this, then I will explain you with the diagram. So now come to the basic structure of our anther. So students, look at this diagram. For any board, whatever board you are following, in that board this diagram is most important diagram. In board, most of the time it is expected by the board to by the students that they should learn it, draw it properly, and most of the time they ask this diagram to draw, and of various marks they can ask it according to their pattern. So look at this. This structure is actually that pollen sac only. The four chambers are there of dithecus, and in between there is a vascular strand. You know that here xylem and phloem is found, and in this portion is called stomium, which is a layer which helps in distance of pollen grains, spreading of pollen grains. So let's start with, we will take this one structure in detail over here. So look at this structure, and the students, you learn it very well and keep all these names on your tips, because it is very necessary and very easiest topic of this plus 2 bio. So make it sure that that should be on your tips. So first layer is called epidermis. You know very well that epidermis is outer layer. It is always helping in protection, so protective in nature. Then next layer is endothelium. Next one, and endothelium is actually it is present below the epidermis, single layer cells only, and mostly it is helping in formation of stomium also. You can see the second layer is stomium is formed by the endothelium only, and this endothelium in the after maturity. After maturity, this stomium will burst and all the pollen grains will come out. So later on we will discuss about this one. First of all, you keep in mind that endothelium is second layer. Now there are 3 to 4 layers of thin walled cells, and these 3 to 4 layers are basically present after the endothelium, and they are actually you can say that sacrificing cells. Whenever any nourishment is required or anywhere any nutrients are required, they will sacrifice themselves. They will give the nutrients from destroying themselves, and then that will be helping for the growth of pollen grains. So next layer is tapetum. Students, most important term is this one, tapetum. This will be asked in all the boards. What is the function of tapetum? Actually, tapetum is having main function, nourishing function. It takes the nourishment from the middle layer and provide it to the microspore mother cell, and by this method the tapetum will help microspore mother cell to develop into a pollen grains. So let's you see the microspore mother cell is lying in between in the center, and they will develop into pollen grain with the help of tapetum by taking nourishment from the tapetum. So tapetum will collect the nourishment from middle layer. So this is all about your this topic, and it is when the anthers will be mature, then student this structure will be formed, this whole structure will be formed, and all the layers will be disintegrate, and only the mmc microspore mother cell will convert into pollen grains. Look at this, pollen grains are present over here, and afterwards the stomium, in the after maturity the stomium will be ruptured, and all the pollen grains will spread outside, and they will spread into the air, and by the various agents they will be taking place to the for the pollination. So students, this is all about your microsporangium formation. And one more thing, you have to basically practice of this diagram and this diagram. These two diagrams must be well known to you, and you just do the practice of these two diagrams. Whenever they will ask you to draw microsporangium, then you have to draw these two diagrams in detail. If you want to explain these all layers over here only, that's also good. You can explain over this diagram only, then one diagram only. But if you will draw like this, then you have to explain this one also, you have to draw this separately. Otherwise try to explain all these terms over this one, then one diagram only you have to draw. So students, hope that this diagram is clear to you. Now I want to, as I told you in the beginning, that this microspore will convert into pollen grain. That process I want to tell you. So let's start with that process, and I will explain you how we can do. So students, now I am going to tell you about the microsporogenesis. Microsporogenesis is the process of formation of pollen grain from the MMC. That means microspore mother cell. Students, microspore mother cell is also called as PMC, that means pollen mother cell. And these pollen mother cells, microspore mother cells, or MMC. Now students, keep in mind, whenever you will go for the NEET exam, then also they will never explain you what are MMC. Most of the time some questions will be asked on this term only, and students will not be able to understand what is MMC or PMC also, or PMC. Otherwise it is very easy term. Microspore mother cell, why we are calling them as a microspore? Students, this is one word, microspore mother cell. Micro means very small. The male unit, that is pollen grain, very small in size, that's why they are named as microspore. The cells who are going to produce microspore, they are called as mother cells. So MMC. Okay, simple one. And next one is, these microspore mother cells will undergo meiosis. That I shown you in the beginning, those that flow chart. In that, microspore mother cells will undergo meiosis because these microspore mother cells, as I told you, suppose this is a microspore mother cell, it is always having diploid number of chromosomes, and then it will undergo meiosis, and it will convert into a microspore. Actually microspore tetrads are 4 cells are formed after this. As I told you that after, as you know that after meiosis 4 cells are formed, and these are always in a haploid manner. Means you can say that these are haploid, and these haploid 4 microspores, these are called as microspore. Okay, microspores. Okay, and this is your MMC. I'm just writing in short, MMC. Now these microspores will never remain separated from each other. They always form a very they will stick together. They form a cluster of 4 cells, and that cluster of 4 cells is named as microspore tetrads. Microspore tetrad. Students, tetrad means 4, group of 4. Tetra means 4. group of 4. So these microspores will cluster together, group together, and they will form a 4. For example, suppose they will group like this, okay. 4 microspore mother cells will group together and they will just join together and they will form a tetrad. So students, there are various flowers are there, various species of plants are there. So different plants are having different types of, they are going to form a different types of tetrads. There are 5 types of tetrads. Number 1 is tetrahedral tetrad, number 2 is isobilateral tetrad, decussate tetrad, T-shaped tetrad, and linear tetrad. Students, these 5 types of tetrads are formed by the microspore, and then these microspore will be going to become a pollen grain. Microspores will convert into a pollen. Later on they will get mature and they will form a pollen grains. One thing I want to just show you, and also I want to tell you that what is the there is some exception that I want to just explain you. One is, there is a word polonium. 1 minute students. Polonium. This word sometime asked in NEET also. Polonium. Actually, in some plants, some orchids or calotropis, the sporangium, pollen grains of a sporangium, they will be not forming any tetrad or any, or they are not getting separated. They just make a cluster or group, or they just make a single mass. They just join together and then they will be forming, that case is called as polonium. For example, students, one example is there, special structure found by the microspore, that is polonium. Look at this. Sometimes all the pollen grains will form this type of structure. You can understand, inside all the pollen grains are just joined together. Otherwise, you know that pollen grains are always separate and simple particles are there, which we can, when dust from the flower, that will be appears as a simple particles only. But sometimes these type of structures of pollen grains can also be found, and this is a special type of orchids are there. Many orchids are showing this polonium type of pollen grain, okay, and they are getting stick together. And one more special type of pollen grains are there. They are also get clustered together and joined together, and the 8 to 64 pollen grains are remain united. Those are called compound microspore. Actually, they are forming like this structure. So many simple structures are there inside and outside. So by this method they are going to form a compound pollen grain. So these two are just exceptions. Otherwise, though most of the plants are forming a tetrads only, and different shapes of tetrads can be visible there. And that as I told you, we can see from this structure also tetrads. I want to show you. Look at this, students. This is polonium. As I told you, exceptional case of pollen grains joined together and they form one disc, one caudicle, and then pollinia. And then pollinia or pollinium also you can ask call it. And this is a compound. All pollen grains are combined together. They will just come out like this only. So this is exception. And most of the plants are showing microspore tetrads. So this is tetrahedral, this is isobilateral, this is decussate, and this is T-shaped, and this is linear shaped. All 4 cells, as I told you, four microspores will be formed from MMC, and they will just make some pattern. In different plants, different patterns will be there. So those patterns are called microspore tetrads. So these 4 are those microspore tetrads. So students, this is what I want to tell you today. Today's topic is this much only, and that I just want to finish it here only, so that you can just prepare it and learn it, all these topics properly. And whatever I told you important, that you should focus on that one, and then rest I will tell you in next video. Thank you.